Thank you for visiting our poster. Today we are talking about the role of C9ORF72 in Purkinje cell function and motor control. Loss of function of C9 protein is one of the main hypotheses of C9 ALS pathogenesis. Knockout mouse model is a useful tool to study the loss of function due to decreased C9 protein and its ability to carry out its still unknown cellular role. We input a C9 complete knockout mouse line from Jackson Lab and assess its motor performance. We found decreased latency to fall in rotarod test and increased sleep in beam walking test, suggesting impaired motor coordination and balance. We further found motor learning deficit in the knockout mice. Unlike wild-type mice, knockout mice had no improvement in trial 2 over trial 1. The gait analysis is widely used to detect cerebellar dysfunction. Knockout mouse showed abnormal gait compared to wild-type littermates. The spontaneous locomotion of mice was evaluated, and hyperactivity was found in knockout mice. We further assess if there is any muscular weakness. Heavier wild-type mice had more strength as predicted. However, such correlation did not exist in knockout mice. The second cohort of older mice showed decreased grip strength in forelimb. Overall, these results suggest that lack of C9 protein is sufficient to produce motor deficits, hyperactivity, and impaired grip strength. Growing evidence indicates the importance of the cerebellum in the ALS pathogenesis to determine whether there is a functional alteration of cerebellar Purkinje cell due to loss of C9 function. We performed slice recording and found a higher filing frequency of the Purkinje cell. Calcium-activated potassium channels like BK and SK are known to modulate Purkinje cell filing. We found the BK channel significantly increased in knockout mouse cerebellum. We further apply BK channel activator on wild-type Purkinje cell and found similar hyperactivity was induced. These results suggest that C9 play a critical role in maintaining normal level of BK channel activity, thereby regulating the filing property of Purkinje cells. We further generated dried mice to chemogenetically activate the Purkinje cell activity. The dried mice with CNO injection showed similar motor deficits, suggesting that hyperactivity of Purkinje cell is sufficient to cause motor deficits found in the C9 knockout mice. Overall, our results suggest that loss of function of C9ORF72 leads to abnormal motor performance and functional change in Purkinje cells, highlighting a critical role of cerebellum and potential contribution of C9 loss of function to the pathogenesis of ALS and FTD. Thank you.